So when you look at question 1, it gives a breakdown. You have a table in front of you. It says germination involves the breakdown of food stores in a seed. Figure 1 shows the results of an experiment where 50 germinating seeds were exposed to varying concentration of pollutants in water. So when you look at the table, it presents on the on the graph, or the table rather, you're seeing the concentration of polluted water in terms of percent. So we have 0, which is distilled water, and apparently they did not place any pollutant in that water. We have 1%, 2%, 5%, 10%, 20%, 50%, 20, 30, 50, and 100% in terms of the concentration of polluted water. If you want to look on the number of germinated seeds, you're seeing how many seeds were germinated when you had the water varying concentration with the pollutants. So where you didn't have any pollutant at all, you probably can just indicate this. We saw where there was actually 50 seeds that was germinated, or that had germinated. All right, so we have 50 seeds. When we look at the 1%, we also had 50 seeds. If you're looking, and I'm just estimating, at let me just double check. So they would have not provided that information. I was just looking if they would have given that to us, but no. So 48, round about 48, right here. Seems like about 45. Maybe 42, 43. About 39. Definitely 30. This is looking like about 21. Zero. So this is the representation of how many seeds germinated based on the concentration of the water. Now it says list four factors rather other than pollution that can affect the germination of seeds. So when we look at germination, We know that seeds, usually we have three factors that we know influence germination. Sometimes in biology, students may want to use the word water, but I would want to say moisture. So availability, the availability of moisture is one factor. So not just to say moisture, but or water, it's the fact that it's available, right? And so we know that water is needed to dissolve the stored food. The food that is stored in the cotyledon would need to be dissolved so that the seed can utilize it. So dissolved food needs to be soluble so that it can basically, the developing seedling can cause this food to get to the area where it is needed. So available moisture, and that would be coming from, in this case, probably the soil. So the soil having in the moisture that it needs. Suitable temperature. And again, seeds will germinate in varying temperatures based on the type of seed. Temperature is needed because when you have that moisture, in order for it to be utilized by the enzymes need to be at a particular temperature. You know, enzymes have an optimum temperature that they will operate. And so once you have this seed at the desired temperature, based on the seed, you know, other little factors that will surround the variety of plants, the type of plant, those things. But generally, the temperature is affecting the enzymes and how they work. So temperature, suitable temperature, is another factor that will basically have an impact on the germinating of these seeds. Oxygen. So available oxygen, and oxygen is needed for respiration. So we would want that to be available. In some cases, you may see them use the term air, but air, the main factor about it, in this case, you really want the oxygen so that the seed can germinate. Remember, even in the air, you would have had carbon dioxide, but the carbon dioxide would not be needed as yet because the plant would not be photosynthesized. You don't have a seedling present as yet. All right, so continuing, and as we said, we're looking at the factors. So depth. Now some seeds, well, you basically have a distance in the soil, like it or not, that you have to put the seed in order for it to germinate successfully. And some may link it to the fact about, you know, you can put maybe in bracket light. So some persons will say that light doesn't really affect the seedling, but the amount of soil, the amount of the distance that you place the seed in the soil will influence if it germinates or not. If you place that seed way down into the soil, it will have implications for the shoots to basically emerge out of the soil. So you should not be having the seed at a certain depth if you want it to germinate successfully. So the depth at which it germinates is also a factor. Even though it says 4, presence of pathogens. Pathogens are able to hamper or disrupt, causing maybe a disease, and it will allow the seeds not to germinate, and you will basically think that the seeds are not good, but maybe there's a pathogen in the soil or somewhere where you're planting the seed that affects the process of germination. So these are 5 factors that you could have listed. Remember, once it says list, you don't have to do any explanation. I basically did the explanation because of a means of using it as a teaching and a revision sort of an aspect, just to look back at what are the things needed for germination. Part B says, state the aim of the experiment. What is it that the experiment, what is the aim? And usually when you're describing the aim, most of the time the sentence will begin, or the statement will begin, by stating to determine. If you can go, if we take a step back, we were looking at the concentration of polluted water. We were all, well, the students or the persons were looking at how many seeds were germinated based on the varying concentration of pollutants. So one way to place the aim is to determine how the concentration of polluted affects germination, right? And bear in mind, you might have other ways of rephrasing the statement to say the same thing. But just offhand, to determine how the concentration of polluted water affects the germination of seeds, could be a sample aim. Using figure 1, explain how the concentration of polluted water affects germination at each of the following concentration. Now, remember, in total, the experiment would have utilized 50 seeds. So when we look at 1, which is right here, we recognize that all 50 seeds did in fact germinate. So what would we say about the percentage right there? How did this concentration, did it affect germination? No. So the 1% at a concentration, this did not affect the germination of seeds. If you want to rephrase it, it didn't have any effect, or it didn't prevent the germination of the seeds. The next concentration would have been 50. And each time, if you're not sure, you go back to the table with the information just to examine. So at 50, and remember we did that evaluation of the amount of seeds that would have germinated just by looking at the numbers. We have, out of the 50 seeds, we're seeing 21 seeds germinating. So it did have an impact. So at the 50%, affected the germination of seeds, causing only 21 seeds to germinate. So some persons may want to go percentage. You can go percentage less than 50% of the seedlings, or sorry, seeds germinated. Part D. Part D says the research team provided the results of the experiment to a farmer. And we know farmers are persons who are cultivating crops. They grow their crops, and this information will be valuable to them. State 2 ways in which the farmer can use the data to improve his farming practices. So we know that the farmer is going to be germinating seeds if he is a crop farmer. So one way it can improve, remember, we're seeing overall, let's go back to the table. We are recognizing that as the concentration of the polluted water, as the water get more concentrated in terms of pollution, it has a negative impact. It reduces the amount of seeds that are able to germinate. So now, if we give that information to a farmer, he can ensure that the water that he's using is of a certain quality, because he now knows water is not just water. So the farmer may have thought before, any water obtained from anywhere, you can just use it and apply it. It's a plant. The plant must can use the water and germinate. So now he sees this information, he would have to ensure that the water he is utilizing for the process of germination should be free from pollutants. That is obvious. So again, it's not because a plant and we can just take any old water and just throw on the plant and it will germinate. No, he would now be able to evaluate. He can do a test, can test the water supply to ensure that it is fit before he uses it to apply it to the soil where he has his seeds, or in his nursery or wherever, before he utilizes it for germinating his seeds. The other thing that the farmer can do, what if he doesn't have any choice? One of the things that he probably can do is to select a particular crop that may be resistant. So you can select a set of seeds that may be or that are resistant to polluted water. So maybe not for all the crops, but if he doesn't have a choice and he recognizes that the water that he has, maybe not 100% in terms of the concentration, maybe 50%, but you might have a type of crop that would be able to withstand that level of polluted water and still germinate, right? So he can do that. I don't know if he wants to take on the expense as well. If he recognizes that we saw where when it was 50% of the concentration, only 21 seeds germinated. So one of the things that he could probably do, which is more expensive, if he wants 100 seeds, then he probably have to plant 200. And that in itself will, take into consideration not all of them will germinate. So that's another approach he might take, but that would be more expensive. It's like you're wasting your seed. So he can take these two approach to improve what is happening with his crops or the germination of his seeds. So part E speaks to, and we have to remember, you see, when CXE gives you an instruction, follow the instruction. Even for this paper, you know, I've been indicating certain things on it, but follow the instruction. It says on figure 1, clearly place an X on the bar that represents the results of the control. So you go to the table and any one of these that you would have assumed is the control, you take your pen and you put an X. No ticky, no underline, no shade, just follow the instruction, we place an X. Sometimes not following the instruction may cost us a mark or two in the exam. And so we place it on the distilled water. There's a follow-up question that says, give one reason for your answer. Now, let's talk a little bit about it. We know there are some variables when we're doing an experiment. We have the control, we have the manipulated, and we have the responding. Before I go to the control, we know the manipulated variable would have been the different concentration of the polluted water. That's what they're manipulating in the experiment. So the change in the concentration, and one of the responding variable would have been the total number of seeds that germinated. Now the control is that distilled water, that set of seeds, the water provided to them did not contain any form of pollutant. And that is just there to ensure that there is any form of misunderstanding when it comes on to what is causing or the implication of the polluted water. So you must have one that does not have any form of polluted water or any form of pollution or concentrated pollutants in the water, so that you can see, use it as a control to compare. All right? So the one that was utilized as our control would have been the distilled water, since it had no trace of pollutants. I don't know who she is, but Mina investigated the growth of seedlings, well, of some seedlings, and made a comparison with growth patterns in a human baby over a 30-day period. The data are provided in table 1. So when we look at table 1, we're seeing the information. At day 5, we have 10 in terms of the height of the seedling. In terms of the length of the baby, we have 45. In terms of day 10, 13, 48 respectively. It says on the grid provided on page 7, plot the data obtained for both the seedling and the baby using the same scales and axis, provide a suitable title for the graph. Now, one of the things that I tell students Sometimes I know this paper is not showing properly in terms of the numbering because I'm incorporated in a Word document. And so you might be in the exam and you have this page on maybe page 5, the graph is on page 7. You can just jot down this data on the page, not on the graph. You can just, jot it down somewhere for your own benefit so that you don't have to be skipping back in terms of back and forth with the graph and the information. It saves on time. Someone asked earlier in terms of what is used. So we basically use our pencil to construct the graph. Now, CXC did not provide any scale for this graph. So usually they provide the scales at sometimes, not necessarily usually, but sometimes they do provide a scale. And so we are going back. Let me see where it is, if it is here. No. So 2 centimeters to 5 days on the x-axis for the days. So 15, 20, 25, 30. So this is days. In terms of the y-axis, that's where we're going to be putting the height or the length in centimeters. This one we can use, it's going to be a little tricky, 5. So 1 centimeter to 5 centimeters. You're placing both heights on the same graph. So at 5, the height of the seedling would have been 10. For the baby, it would have been 45. At 10, it's 13.
I did a graph like this before, but it come out like that. I thought it was weird, so I thought it was wrong, but it's the same thing. So far, it's on track at what I did.
Wonderful. 48. At 15, it's 16. And then we have 52. We're at 20, 20. And then 58. We have 25, 25. And then 61. And then finally we have 30 at 30. And of course 64. Now bear in mind I'm utilizing the computer. So it's not so 100% in terms of accuracy, but we would have gotten the idea as to the plotting. So you would have labeled, this would have represented, so we would have completed the curve, as I said before, and no question regarding a title was also a part of it. Well, a part of the question was for you to provide a title. And normally, we start by saying graph showing. And basically, you just explain what is the graph showing. The growth of a seedling and a baby over a 30-day period. Right? So that's a simple title to have. Remember, because of how I was doing the input of the numbers, you would have still labeled this axis with the height slash length in centimeters for the baby or the seedling. That would have been needed right there. So if we go back, we would have completed just doing that, would have provided you with 6 marks. So again, persons ask, should I use a pencil or a pen? If you use a pen, you already know the disadvantage of that. So that would, and you know how we are presented with these in terms of a booklet. You're provided with one booklet. Not that you may not be able to get a second graph, but use the pencil, ensure you have your eraser, ensure that you, if you make a mistake, you erase clean. Now part G speaks to suggesting two ways in which the growth of the baby differs from the growth of the seedlings. Now we know that seedlings, referring to the seeds, they get their nutrients initially from the stored food, which is the cotyledon. So they get their stored food from the cotyledon, and so that would have been utilized for the initial parts of their development. For the baby, the baby would have obtained nutrients from the mother, or well, if you want to, when you're thinking from the mother, we're really referring to the breast milk or any other nutrient rich food. So whether, baby feed. So that is where you have that difference in terms of how they obtain their nutrients for growth. The next thing is that the seedling would have some level of independence. So when you think about a seedling, once all factors are good, you don't necessarily have to be with the seedling to ensure that it is like you're feeding the seedling, because they have some level of independence. While for the baby, you would have to provide the food for the baby. So you'd have to assist the baby in terms of nutrition. So while you're feeding the baby, or you can just, even if you mix the formula, you would have to ensure that you provide some level, take the baby, the mother would have to put the baby on the breast, things like that. Once you have the seedling, the seedlings have some form of independence where once the nutrients is available and they grow their roots, they grow their shoots, they just take up what they need from the soil, and that would be it. So you don't have that constant attention like what is required by the baby. So part 2 says suggest one similarity between the growth of the baby and the growth of the seedling. Now when you think about, and I didn't talk about this earlier, when we refer to growth, we talk about a permanent increase in the size, right? And so not necessarily that the organism increases, but it's permanent. So one similarity is that both organisms will undergo mitosis. To increase in size. So you have we have the term cell elongation and cell enlargement, and both are through the influence of mitosis. So when you think about the plants, the enlargement, when you think about the terms of the baby, we talk about the elongation of the cells, and both of them will allow for an increase in the size of the organism. It says, part H says, describe how the seedling height is measured using a ruler. So one way, if you have the seedling, of course, you're going to be placing the ruler at the base. If it is that the seedling is in the soil, and you would have aligned the ruler vertical from the point at which it starts at the soil to the top of the shoot, and you read the length from the ruler and record it. Due to the influence of time, so you basically place the ruler at the base at where the stem of the seedling would have been emerging from the soil, and you would have aligned the ruler vertically so that you can record the height to where the shoot would have stopped on the ruler. So just one example that shows that an increase in cell is not always due to growth. And I just made mention about not all the time when something increases, it means that it grew. So growth is a permanent. And so for this, osmosis is a pretty way to basically explain this concept of increasing in size. And so you can use an example that you know in regards to osmosis. So if you talk about potato strips, so potato strips being placed in distilled water, so they will increase in size due to water entering the potato. But as you know, if you take this very same potato and place it in a brine or salt water, it's going to reduce in size. So that size is not something that is permanent. So this is not the cells, sorry, this is not due to a growth in the potato strips. The potato strips did not grow. They increased in size, yes, but it was not something due to growth, or it wasn't permanent. And so this is a prime example. As I said, osmosis is the best, one of the best examples you could use. Remember, you have some organisms such as predators that will, like a frog. A frog will, swell up just to try and get larger than another or to ward off predators, and they increase in that size, but it's not a permanent. So that's another example that you could have used. Remember, potato, papa, whichever one you want to utilize in terms of osmosis. It says, suggest one factor that could be considered before making a conclusion from the data. And if you're looking at the data, we are seeing information for the seedling and for the baby. But one thing you would have to take into consideration is the type of plant. Because varying plants, if this plant only grows to a particular height, or if this is a permanent, like a mango tree, then this is not a true reflection of the entire growth, period. Some plants will grow in a particular direction as well. Some are utilizing vines. So one thing that you should take into consideration is the type of plant that you're comparing this growth of baby to. Cool?